Hi, good evening, everyone. Sorry, I'm a little late today. So I will right away start the session. So we will again summarize week three uh, content here. Uh, yeah. All right, I will start sharing the screen. Mhm. Okay, someone wants to join the call. So once I start sharing, I can't see who wants to join the call. You guys can unmute yourselves and let me know if someone wants to join the call. My screen is visible, right? Okay. So, today we will be summarizing the week three content, uh, where it was basically about starting of how are you making genetically modified organisms. And the content will also have cloning, about cloning, DNA isolation, both plasmid as well as genomic, and how do you really, the whole process of cloning, we will look at it in some detail. Okay. So now when we talk about genetically modified organisms, which means that at the genetic level or the at the DNA level, the nucleotide or the gene sequence encoding for RNA or encoding for proteins are tweaked or changed so that we get our phenotype of interest, or we get basically to perform any function of our interest, or something of that sort. So for example, for production, for industrial purposes, what we looked at for different biotechnological applications. So the organisms which can be genetically modified can be uh, bacterial organisms, plants, animals, viruses, etc. Okay. So now what happens here is, um, this is an example of plant, genetically modified plant. So what happens is, suppose, what is the general flow of preparing one genetically modified organism is, suppose a particular pest is there, plant pest is there. We need to stop that pest from infesting the plants. All right. So what happens is, suppose some kind of gene that is harmful for the insects or for the pests is encoded by a certain gene. That particular gene, insecticide gene, is inserted. Okay, inserted into, this is the green one is the particular gene which encodes for the uh, chemical substance, or encodes for RNA, any kind of substance which is harmful for the pest. And what happens is, we basically cut it out into smaller parts, because we, this is the whole DNA. Now this part is only of our interest, this particular sequence is only of our interest. So what we do is, we have to take it out of the whole long sequence of DNA. So we chop the DNA into small fragments, and then what happens is, now these DNA, this is from some other source. Now this particular genetic sequence which encodes for the insecticide, okay, uh, that has to be uh, carried into the plants, right? So that the plants can produce that insecticide, so that the pests or insects can't feed on the plants. Okay. Now what happens is, so that has to be carried into that organism, that has to be introduced into that organisms. So that carrier, or it is also called as vector, are these plasmid circular DNA molecules. Okay. So what happens is, this is the gene of our interest encoding for the insecticide, which is joined or fused with, or the correct term is ligated. Okay, we will look into it later. It is ligated with these vectors, plasmid vectors, which are also cleaved, because otherwise these can't really join. Okay, thereby forming these recombinant vectors, recombinant DNA. Okay, and then these DNA are taken up by the plant cells, okay, or the plants, and then definitely these now, whether this particular vector along with the gene of our interest has actually gone into the plants, that for checking that there is some kind of selective marker always present. Okay, so that out of, otherwise if there's no selection, whether the plants, any kind of plant, as in I mean any kind of plant, whether it has the recombinant DNA or not, will grow on the nutrient medium, but that nutrient medium has to select for this particular, specifically for this particular recombinant DNA, so that only those plants with this particular recombinant DNA can grow. Otherwise, like all other plants which did not take up this particular recombinant DNA won't grow. Okay, now these then plants are grown, and what happens is now all these plants which have grown here on this nutrient medium have been selected. That means these have these recombinant DNA inside them, and then this green part is the particular gene that is also, that has also been inserted into the plants. And then what will happen is these plant cells will produce that insecticide, because it now has the gene which encodes for the particular insecticide, and when the insects will feed on the plants, they will eventually die. Okay, so this is just an example of how what is the basic workflow of producing one genetically modified organism. Okay, now let's look at some history. That in very first for the very first time in 1973, Boyer and Cohen, they actually inserted a kanamycin resistant gene from one bacterium into a plasmid of another bacterium. Kanamycin is an antibiotic. Okay, so from one bacterium, we looked at in the previous slide, there were circular DNA molecules, plasmids. Okay, so one kanamycin resistant, how in that, in this earlier part we have this green gene which encodes for the, I mean green colored segment which encodes for the insecticide is inserted, okay, into the plant cells. Similarly, for the very first time in 1973, Boyer and Cohen, what they did, they inserted one gene from one bacterium into another bacterium, and that particular gene was uh, helping the bacterial cells to be resistant to a particular antibiotic, which was kanamycin. Okay, so when this gene got transferred into another bacterial cell, it transferred the kanamycin resistant trait. So this particular, this is the first instance of GMO or genetically modified organism developed by humans. Then again in 1974, they only transferred a gene from Xenopus or frog into a bacterium. This is again an example of first genetically modified organisms with genes from another kingdom. Okay, this is like from bacterium to bacterium, and this is from Xenopus into a bacterial cell. Okay, and then in between 1977 to 78, genetically modified E. coli. E. coli is a bacterium. Genetically modified E. coli species which encoded human insulin hormone was developed by this particular company, GeneTech. So because, and it was named as humulin, this E. coli produced insulin was named as humulin. Okay, and then finally in 1982, FDA approved the use of this humulin or E. coli produced insulin, and this humulin is the first product from a genetically modified organism to be used for consumption. All right, this is just a history, like from when did actually these cloning and genetic modification started. Production of genetically modified organisms basically started. Now there can be, this is from the lecture slides only. So there can be different approaches for isolation of gene of interest. One is that you don't really know the gene sequence at all. Okay, you want to insert a particular genetic sequence of your interest. You know that, suppose in that example of insecticide gene insertion into the plant cells. Okay, now you really don't know which particular gene sequence encodes for the insecticide. Okay, so in that scenario, either what can be done is, let you can use the genome to form a genomic library, or you can have the transcriptome, basically all the encoded RNA, and then reverse transcribe it to form the DNA, which is called complementary DNA. And then you screen and then have complementary DNA. And then similarly, if that particular DNA is able to you know pass through some selective markers, we will all look through it, go through in the subsequent slides. Then we will screen through it and then select our gene of interest. Now if the gene sequence is known, we already know that okay, this particular sequence of genetic sequence encodes for our insecticide, uh, for the insecticide. Then what happens is, you have the gene sequence already known. You basically, now the thing is, suppose you have only one copy of that particular gene which encodes for the insecticide, but only one copy won't be enough. You have to suppose insert it into numerous plant cells. Okay, so you need a larger amount of that particular genetic sequence. Okay, so you basically amplify it. Okay, the particular experiment or the technique is used is polymerase chain reaction. We will look at it also. And then you get that amplified DNA fragment, and then you use it for insertion into plasmids, and then the recombinant DNA to make recombinant DNA, and that recombinant DNA is inserted into the plant cells or host cells. Now, cloning actually describes different processes that can be used to produce genetically identical copies of a biological entity. Okay, basically to produce clones or similar identical copies of, now in molecular cloning, it's producing genetically identical copies of a biological entity, or um, for biological entity, how you are basically taking the genetic sequence which encodes for that particular entity or particular substance. Okay, now we looked at the first part where we had to, when the genetic sequence is not really known, we can construct a genomic library. How would you do that? All these terms we will eventually come to it, but very to give a broad overview, it's like first you have to take out the whole DNA content, whole genomic content. Okay, and then you chop it off because you need to have your um, genetic sequence of your interest. Okay, and then you definitely, after chopping it off, after digesting it using restriction enzymes, so the particular enzymes that are used to digest genetic sequences are called restriction enzymes. And then after chopping it off into multiple fragments, you purify those DNA fragments, and then your you ligate those fragments into an appropriate vector. We will come to all these vectors. This is an yeast artificial chromosome vector, YAC vector, and then you transform into your host organism. This is an example. So the host organism here is yeast. Okay, so how are you creating this genomic library? You will have all these uh, DNA fragments. Different fragments are ligated into a suitable vector, and then that particular recombinant vector or recombinant DNA is transformed into yeast or the host organism. Okay, thereby creating multiple, thereby creating a genomic library having multiple recombinant genes or DNA. Okay. Now, how do you really isolate genomic DNA? Okay, uh, if we look at the brief overview, what you have to do is, DNA is definitely inside the nucleus, enclosed inside the nucleus. So you have to first break the cells open, which is called lysis. You have to lyse the cells. Usually it's done using some kind of detergent. Okay, and the solution inside which you are lysing the cells, you resuspend the cells and lyse it or break it open, is called lysis buffer. Okay, that should have an appropriate pH, which is which is amenable for all the biological entities inside that particular cell that you are about to lyse or break open, should be stable in that particular pH range. Okay, so that these buffers have an ideal pH range which needs to be maintained while breaking open the cells. And then what you do is, you have to digest it using different kinds of enzymes, because you are isolating DNA, so you don't need proteins and other contaminants. Okay, so you digest it, and then you actually pellet out your DNA content. Okay, and then you purify the DNA, and then again you pellet out the um, pure genomic DNA, which you get after all these steps. Okay, this is very quickly I went through it, and eventually in the next slides we will go through each of the steps, what happens in there, why are you using this buffer, what kinds of buffer have should be used in these isolation procedure, that we will go through it. Okay, now when we talked about the very first thing of isolating any kind of macromolecule from it, like this is an intracellular content. Okay, so to take the intracellular content out, the very first thing is to break open the cells or lysing the cells. Now cells can be lysed using different kinds of procedure. One is a very conventional and old way of doing it is mortar and pestle. What you do is, it you have the cell pellet. We looked at in the previous tutorial that you have the whole cell suspension, you pellet down the cells, and then you have the cell pellet there. Now what you do is you put in the lysis buffer, and then you simply, like, you have this thick pellet, you resuspend it or grind it. Okay, give you grind the cells, and this particular mortar and pestle method is used for plant samples which are frozen in liquid nitrogens in the since the specimens are comparatively bulky. Okay, what also happens is, so after the lysis step, there can be different metabolites which come out of the like after the lysis, after lysing the plant cells. Metabolites and samples then can be extracted by adding suitable solvents. Okay, solvents or buffers. All right, and then this mortar and pestle grinding the old cells, grinding the cells, the plant cells, frozen cells or cell pellet, can be used with all types of cells, and also can one can add certain types of beads. So beads can be glass beads, okay, which are like spherical beads which are required. Suppose uh, once you have the cell pellet, you have resuspended the pellet in the buffer. Now these beads, these glass beads, can be added to the resuspended solution, because resuspension is helping in lysing the cells definitely, but it needs to break open also. Suppose the cell wall, suppose if we take the example of plant cells, it has a cell wall to break, and then cell membrane has to open up. Okay, so what happens in those cases, you have the resuspended solution, resuspended cells in the lysis buffer, you add the beads, glass beads basically. Now you vortex it. Vortexing is uh, in vortexing, what happens is, you have a tube, and it you know like, as the literal meaning of vortexing, it's like it centrifuges at very high speed. Okay, and completely you have this tornado formation, right? So how it looks like, so it is completely a spherical circular rotatory motion, and then the whole solution moves in that way. So if it's a tornado forming inside the tubes, all right, in a very plain manner. So what happens there is, you have the beads there. Now the beads are also um, colliding with the cells a lot, and that helps in breaking the cells up. Okay, uh, all right, this is mortar and pestle, and then you can add beads. This is also called as bead beating. Okay, after you have resuspended the cells, so glass or ceramic beads are used to crack open the cells. Again, mortar and pestle bead beating is a kind of mechanical shear. Okay, but this doesn't really harm the organelles. Organelles are usually intact after this bead beating method. Next, you have sonication, which is also very routinely used in protein labs for taking out proteins. Okay, also ribosomes, etc. So what happens in sonication is, again, it's a device or instrument where it has a probe. Okay, sonicators induce rapid vibrations in a titanium probe, which is immersed in the cell solution. Very first thing, what I was telling earlier, that you have the cell pellet, you have resuspended it in the lysis buffer, and then this particular probe is immersed in that resuspended solution. Okay, and when this is immersed in the resuspended solution, then cavitation happens. Okay, when the instrument is switched on, it uh, sonicates in pulses. Okay, when it has an on time as well as an off time, like off on duration of few seconds, then off duration of few seconds. Okay, so what happens when it's on is actually cavitation. What happens is tiny bubbles are produced within the cell suspension, and then bubbles explode. So what happens is local shock wave, like uh, passes through the whole cell suspension, and that actually helps in disrupting the cell wall. Okay, um, medium is liquid. Okay, and this cavitation thing is almost like an explosion, which usually disrupts the things in the blast radius. All right, and medium is liquid. Just in case of normal, when we have bomb explosions, it's air usually, but in this scenario we have the medium as the liquid medium. All right, um, and then homogenizers. Okay, another way to lyse the cells is the usage of homogenizers, which again helps in, like, almost it's mostly like the bead beating method, where again mechanical shear is used to break the cells open. And also what happens is, suppose you have a layer inside and the cells are squeezed. Okay, and the particular layer has a gap which is lesser than the dimensions of a cell, one cell. Okay, so if the diameter it is less, the particular gap is actually less than the diameter of the cell. So when the cells are passed through that particular gap, what happens is it will remove the, or it will create a shearing force which will help in removing the outer layer of the cell. Okay, so it's basically very less, the gap is like really less, because in the orders of um, what the cell's diameter is. Okay, so homogenizers are again used for mechanical shearing, for producing mechanical shear, which helps to break the cells open. Okay. Then there's a high temperature lysis. What happens is high temperature and pressure break the chemical bonds within cell walls, and it also denatures proteins. Especially high temperatures can denature proteins. So, but what happens is it is like really quick. You are um, like it's a really fast way to break the cells open, but it's a little dirtier thing where you will have, like, you basically microwave or autoclave to generate the heat and pressure. Autoclave we looked at in the last tutorial also, but this should be avoided in case when the sample is denatured by heat. Okay, when I was talking about protein prep. Okay, during purification of proteins are usually sensitive to, many proteins are usually sensitive to high temperatures. So heat actually denatures the proteins. Even this particular denaturation step is also, this particular high temperature denaturation is also used for um, preparing protein samples for electrophoresis. We will look at it in later tutorial videos. Um, so there what happens is you are basically heating the protein up to denature any kind of structure that they have. The whole structure of a protein is basically due to different intermolecular forces between them, right? So what happens is high temperature breaks those bonds and helps it to linearize the proteins. All right. But suppose if we want to purify proteins, so this particular high temperature lysis method should not be used to break the cells open. Okay. There can be enzymatic lysis also. It breaks like lyses the cells using enzymes that degrade cell walls and membranes. Okay. So depending on the type of organism, one can use cellulases, right? Chitinases, then bacteriolytic enzymes like lysozyme, which destroys the peptidoglycans, mannases or glycanases. If you want to suppose isolate a protoplast, which is actually a cell without the wall, you can use enzymatic lysis to remove the cell wall. If the particular cell of interest has one, you remove the cell wall and the cell membrane. Okay. And protoplast actually is only the cell without the wall. Okay. Then there can be chemical lysis, as the name suggests. You use chemical agents to remove the layers and take the content, cellular content out. So there can be different organic solvents like alcohols, ether, or chloroform, which disrupts the cell wall by permeabilizing the cell walls and membranes. Okay. So suppose if you want to purify plant pigments, so what you do is you extract, which are like hydrophobic molecules. To extract these, you can use these organic solvents. One can also use EDTA, ethylene diamine tetraacetic acid, which is actually a chelating agent. It chelates divalent ions. All right. So what happens is many times the divalent ions like um, magnesium or calcium helps in stabilizing the contents of the cell wall. Okay. uh, as in the case of gram negative bacteria, the cell walls contain lipopolysaccharides, which are actually stabilized by magnesium and calcium ions, divalent cations. And when EDTA, which is a chelating agent, it chelates these divalent ions and then destabilizes the cell wall, and then you can remove the EDTA later. And why do we usually have to remove the EDTA later? Because many times, not only the cell walls, suppose many proteins or in general the cellular contents also have divalent ions or many kinds of ions to help, which help perform their functions, which help stabilize their structures, etc. All right. So if EDTA still remains in the particular lysis solution or lysed cell, uh, like lysate, okay, so in that case it has to be removed, otherwise it will chelate out all the other ions, which will eventually harm your sample of interest, whichever molecule, suppose proteins. If you want to purify the proteins and you are dialyzing, I mean you are still having EDTA in your solution, it can help, it can harm the structure of the protein as well as the functionality of the proteins that you want to purify. Okay, because many times protein structures, etc., are stabilized by certain ions. Okay. Then another is freeze-thaw cell lysis method, which actually, if, like freezing creates ice crystals and water expands upon freezing. And when the sample is thawed, um, ice crystals eventually melt. So multiple freeze-thaw cycles helps in rupturing the cells, and which is comparatively gentle. Definitely doesn't really generate much heat, but uh, it takes some time, like quite a bit of time. It is time consuming. So usually it's not used in a routine um, isolation procedure. Okay. Now, Um, another way to purify DNA, in general, DNA, RNA, and proteins, one way to do is phenol, using phenol chloroform extraction. So what happens here is phenol is added to the aqueous soup containing the proteins and the DNA to be purified. Okay, so you have centrifuged, you have lysed your cells now, you have the cellular contents out. Now, what you do is you centrifuge it to separate out the cell debris, etc., which will pellet down, and the other cellular contents will remain in the supernatant or solution, which is what is referred to as aqueous soup here. Okay. Now, what happens is phenol is added to that particular supernatant. Now, that aqueous supernatant and phenol are actually immiscible and do not form a homogeneous mixture. Now, what will happen is upon adding phenol and mixing it properly, it because it's immiscible, you will see like very distinct phases. One is the aqueous top phase. It usually top phase is aqueous phase and phenol phase. Okay, because phenol is denser among the two liquids, so it will stay at the bottom. Now, also usually it should stay at the bottom, but due to vigorous mixing and all, what also happens is sometimes the layer flips. Okay, so that should be avoided. Okay, when you really want to purify DNA, which is usually in the aqueous solution, DNA or nucleic acid, which is usually in the aqueous phase, if it flips, then it becomes difficult to take the nucleic acid or aqueous phase out. Okay, so we should be gentle while mixing. uh, and so if we mix it properly, then phenol being denser out of the two liquids, which are like phenol and water here, it sits at the bottom, and then phases are mixed thoroughly. What happens is this mixing helps phenol forces the phenol to move into the water layer, and it forms an emulsion of droplets. Now what happens is proteins in water phase is denatured, and usually the denatured proteins actually move towards the phenol layer. Okay, phenol layer, and DNA or RNA also, nucleic acid, stays in the aqueous phase. Okay, now yeah, uh, now one thing is, uh, why are why are proteins getting denatured in the emulsion of droplets? Okay, and then it moves into the phenol layer. Why is it so? So there are three scenarios. One is like DNA is usually polar. All right, it has negative phosphate groups. All right, and it has nitrogenous bases and all those stuffs. So DNA is polar, but protein is less polar. Okay, now what happens is proteins flip inside out. In general, in proteins are composed of amino acids. All right. Now, amino acids do have both hydrophilic as well as hydrophobic moieties, right? It has like COH group, etc. But then again, it also has CH3, C2H5. All these moieties are also there. Okay. So overall, it is less polar, usually less polar than the DNA molecules. Now, but in general, if we talk only about proteins, it has both hydrophobic as well as hydrophilic moieties. Hmm? So hydrophilic moieties will tend to uh, move towards the polar side, or polar, more like towards the polar side, and like hydrophobic, less polar ones will try to stay in the hydrophobic medium, right? So what happens is proteins flip inside out because the less polar residues, which are usually hidden inside, because the more polar are towards the aqueous medium, that are hydrophilic, right? Water loving species. So so those water loving moieties are usually outside, and whichever is the hydrophobic moieties or less polar moieties are usually hidden inside. But when it's an aqueous medium, um, or if usually this is the scenario when it's an aqueous medium, but when it's comparatively less polar, phenol medium, which is definitely less polar than the aqueous medium, what happens is now those hydrophobic residues are able to interact. All right, they no need to hide any longer, they can come out of the structure, move, like they can flip outside. Now what happens is polar residues on the outside of the proteins made them soluble in water, and the phenol induced folding changes forced the phenol favoring residues towards the outside. That is what is the same thing that is being said here. Now the proteins are more soluble in the phenol phase rather than the aqueous phase. Okay. Now, some very polar residues may flip to the inside of the globular protein because now the medium is comparatively hydrophobic, okay, or less polar. So, if there are like really polar residues or hydrophilic residues in the protein, then it will tend to flip inside and come towards, like, tend to hide away from the hydrophobic medium, okay. So, what happens is when it's a globular protein, some very polar residues can flip inside, which so as to cover themselves from this unsuitable new solvent, which is the less polar solvent. So in short, the proteins are permanently denatured by this new solvent environment, which is provided by the phenol uh, liquid. Okay, phenol. Now What happens in phenol chloroform extraction is it is a buffer saturated phenol chloroform usually used in 1:1 ratio. Buffer saturated means the pH is maintained at a um, uh, particular range where the I mean nucleic acids or proteins are uh, able to retain their functionality as well as the structure. Now, phenol and chloroform are usually used in a 1:1 ratio for DNA purification, with other ratios sometimes used for RNA purification as well. And many times isoamyl phenol phenol, chloroform, as well as isoamyl alcohol is also used as an antifoaming agent, but it is usually optional. But definitely phenol and chloroform are there. Now, what happens is density of buffer saturated phenol is little higher than water. So, if the aqueous phase contains enough salt or any other solutes to increase its density, then what happens is phase inversion. What I was talking about earlier, aqueous phase comes under the phenol phase. Okay. Um, so, for to prevent this particular phase inversion, chloroform is added, which is significantly denser than water. Now, adding it to the organic phase or the phenol phase increases the overall density of that particular phase, and adding chloroform actually prevents this phase inversion. Okay. And also chloroform addition helps in reducing the interface. What happens is, we talked about in the previous slide that there is an aqueous layer and a phenol or organic layer. Okay, so you can see a distinction there. But um, if many times there's a fuzzy interface in between. So to reduce that fuzzy interface, adding chloroform actually helps in this regard. So the fuzzy border between the two phases um, are is there. Okay, but usually it helps reduce the or rather uh, decrease the thickness of that particular interface. Usually in the interface, what all molecules reside, those molecules which really can't decide where they want to go. Okay, whether they want to go to the aqueous phase or to the like completely denatured proteins are in the phenol phase. Okay, when we looked at just in the previous slide how the proteins are denatured. But suppose um, some like a big protein molecule hasn't denatured completely, so it will stay like it is not able to really decide where to go. Okay, like DNA binding proteins or which are partially denatured, not completely denatured proteins can also stay in that fuzzy border, which is also called the interface. Okay, and fuzzy border, what can be the reason for this fuzzy border? This is due to the molecules which can't decide where they want to go, mostly partially denatured proteins. DNA can also be there when we don't maintain the pH, then it won't really love to go to the aqueous phase. Okay, um, or partially denatured DNA binding proteins, which are also bound to the DNA. So those ones, DNA has moved to the aqueous phase, but the DNA bound proteins can't really move to the aqueous phase, but also the binding is so tight that it can't really move to the completely detach and come to the phenol phase. In that scenario also, sometimes these DNA binding proteins can stay in the interface. So also now DNA has moved to the aqueous phase. Now suppose you are interested in purifying DNA. Now you have to very carefully take the aqueous phase out. Now these are usually in small tubes. These mixing, these extraction are usually done. So what you do is you not necessarily always in small tubes. It can be larger tubes as well. But basically, what you have to do is you have to very carefully take the aqueous layer out, because you even if the pipette tip touches the interface, or it should not really move to or it should not really move to the organic phase or the phenol phase as well, because it will contaminate your DNA sample. It's really pure there, the aqueous sample is quite pure. So in order to avoid any contamination, what to do is you take it out very slowly. Also it has to be slow, because if you put in like take it out with a force, it can also suck in the other liquids or the other phases as well. And also, now uh, if we look at, see, this is the aqueous phase and this is the phenol phase. Now, if you want, you are like, okay, maybe suppose 10 microliters are pipetted out, and still you can take in like 50 microliters more, you are still letting it stay in there. So, in that case, what will happen is you are pipetting out, taking out less volume of aqueous phase. So, it will also lead to a lower yield of your DNA. Okay, so there will be compromise on the yield. Now if you are taking it more than what is required and it's like about to touch, or you are touching the pipette tip to the other layers, then it will decrease the purity of the sample. Now uh, oxidation of phenol, like we have looked at in our 11th and 12th classes, chemistry reactions, that phenol oxidation produces pink or brown oxidation products, and these products have been found to actually degrade DNA and degrade RNA and cut or excise DNA fragments. So mostly the commercial phenol solutions contain an antioxidant which inhibits this particular oxidation, and phenol buffered at an acidic pH has been seen to be resistant to this oxidation. Okay, phenol, like maintaining a particular pH which is usually towards the acidic range, has been found to be helping in reducing uh, has been found to help in reducing this oxidation of phenol. So it's good to move a portion of the buffer saturated phenol from the brown bottle that it likely came in to a clear bottle or tube to inspect it before extraction. Okay. If we see pinkish or brownish tinge, it is not a perfect fit to use for um, DNA extraction or other kinds of like phenol chloroform extraction method, because it can eventually um, affect your sample DNA or RNA. And phenol chloroform extractions are amazingly efficient. Actually, all like um, it's heavily and very commonly used in all kinds of RNA and DNA extraction processes, purification processes, and less than 1% of the average protein remains in the aqueous phase after the first extraction has come to equilibrium. Now, for equilibrium, what happens is more surface area there is between the two phases, the faster it happens. Surface area greater for fine emulsion, and which can be achieved by vortexing the phases for a couple of minutes, but also not all the samples can be vortexed. Now, if we want to purify like very large DNA fragments, like genomic DNA are like very large DNA fragments, so we have to mix the sample more gently, and therefore each extraction takes much longer time because we don't want to shear the DNA. Vortexing also can shear the DNA fragments because genomic DNA are like really large, so we don't want to vortex it. We have to handle the sample much more gently. Now, now you have your DNA fragment, okay, DNA fragment of your interest. Now it has to be finally cloned into a suitable vector, okay. And vector is basically a medium, a vehicle or carrier to carry that gene of interest into a host organism, okay. Now usually the suitable vector to prepare the genomic library is selected based on what is the size of your gene of interest, okay, and what is how much can be basically loaded onto the carrier or vehicle, okay. Size of an average fragment can be calculated, and accordingly a suitable vector can be chosen. In the case of fragment generated by restriction enzyme, vector can be digested with the same enzyme and put for ligation or joining to get the clone or recombinant DNA, okay. In the case when mechanical shearing is applied, these fragment needs additional effort. What happens is, in one of the approach, an adapter molecule can be used to generate sticky ends. Alternatively, an endonuclease can be used to generate sticky ends. Sticky ends are basically, I think, we have an preliminary knowledge from our high school studies that uh, DNA can have fragments can have both blunt ends as well as sticky ends. Blunt ends can let the DNA join in any direction, but sticky ends helps in a proper frame in joining of the complete recombinant DNA, and the reading frame remains the same. Okay. Now what you do is you have cloned it, you have the recombinant DNA molecule, you have to put or introduce that particular recombinant DNA molecule inside your host cells. Whichever host molecule or host cell you want to have, whether it be it plants, yeast, bacteria, mammals, you can transform it. Okay. Now, clones are transformed in a suitable host to get colonies. A suitable host can be a bacterial strain or yeast. It can be mammals also. Usually we use the term transfection in those cases. And different methods are used to deliver the clones, which will eventually be covered in the further slides. Not today, I think the future tutorial videos. Now, the carrying capacity, like the particular depending on the size of the fragment, as well as the size of the carrying capacity of the vectors, um, how much of the DNA size or genetic sequence can be inserted. Okay, so yeast artificial chromosomes can have like 250 to 2000 megabases of insert. It's like really large. That's why this is used in what we looked at in the very first example of creating genomic libraries. It has like the whole genomic DNA is chopped into multiple fragments. So it has like really bulky fragments to ligate and form recombinant um DNA. And that is actually the carrier in those cases are East YAC vectors, what we saw in the very first few slides. And then there's bacterial artificial chromosome as well, BAC, which can have an insert of 120 to 300 megabases. Okay, and then plasmids usually can have 15 megabases of insert. So this is what we looked at. Okay, creation of genomic library. And now one way was to have genomic library creation, and another is to have the left DNA like gene sequence or DNA sequence, um, transcribe the mRNA, and then from mRNA you reverse transcribe it to form the DNA. So what happens here is, uh, again you have the tissue, you homogenize it, and then you isolate the mRNA from there, and then after isolating the mRNA, you have to construct the cDNA. So you have the mRNA, you collect it, now reverse transcriptase to form the DNA, which is complementary DNA. Now this particular DNA is inserted into the plasmids, and these plasmids to form the recombinant plasmid, and this recombinant DNA is inserted into host cells, and the ones and these cells are selected for to grow, to allow the growth of only those cells which have taken up the recombinant DNA. And then the plasmids recombinant DNA are purified in much larger quantities from those host cells which have taken up the recombinant DNA. And then in order to check that okay, there is no mutation happening or there's no error in the sequence, what one does is, it's always a good idea to check it via sequencing, so that you know that okay, this is the exact gene sequence or DNA sequence that you have. Now this is where after sequencing, in this case cDNA library, what we saw earlier, that cDNA library is done when gene sequence is not known. So you have the cDNA library now, and you can know the sequence directly. So this is what has been shown here, that you isolate the mRNA, you prepare complementary DNA fragments here, and then you clone it in a suitable vector system here, and then transform into a suitable host, and then purify the plasmid finally for storage, and also get the sequence checked via sequencing. So complementary DNA or cDNA is an extremely useful tool which has been a gift, which has proven to be a real gift to molecular biologists by RNA viruses, which fortunately invented the reverse transcriptase enzyme that allows us to make this stuff. Okay, this reverse transcription mechanism, the central dogma used to be unidirectional, right? Now, due to these RNA viruses, we have the process of reverse transcription as well, from DNA to RNA, but RNA to DNA can also be made using this particular enzyme, which is actually very commonly used by RNA viruses. RNA viruses use this mRNA dependent DNA polymerase. Reverse transcriptase is actually an mRNA dependent DNA polymerase which converts their genome into double stranded DNA for integration in their host's genome. This is the machinery used by viruses for infection. But in the laboratory, for experimental purposes, it can be harnessed to make cDNA copies from mRNA templates. Okay, so you have like purified reverse transcriptase enzymes commercially available, which can be directly added to make complementary DNA copies from mRNA templates. That means that reverse transcriptase can be used to synthesize a cDNA copy of every mRNA that is present in an mRNA extract. Imagine how wonderful this particular tool is. Okay. So now you have the mRNA, which is usually having a poly A tail. Okay. Now a complementary thymine containing residues, this particular oligo DT primer, which is having around 20, which is having 20 nucleotides. This is uh, synthesized, and this actually binds to the poly A tail. And then what you have is you put in reverse transcriptase enzyme, add a like in a the reaction condition has to be taken care of in terms of the temperature where the enzyme is has the optimum activity and the duration. And then you also put in, definitely you have to put in the nucleotides here, which will eventually, because from the nucleotide pool only it will this enzyme will add. This is basically a DNA polymerase, right? So it will add from this nucleotide pool the residues complementary to the mRNA residues here, and it will synthesize the first strand. Now you don't really want your mRNA. When you already have the cDNA here, you add in an RNase enzyme which helps removes the mRNA, and then you have this first strand cDNA present. Okay. Now only one strand of cDNA won't really help what we saw. It is like a very less in number. So only suppose one molecule is there. Okay. But you have to amplify it in like millions of copies so as to have a huge amount of cDNA formed. So that's when the amplification reaction or PCR comes into picture. Okay. This is another conventional cDNA synthesis that has been shown where the poly A tail of the mRNA is used. Same way the reverse transcriptase enzyme is also added. Okay. Now advantages of complementary DNA over genomic DNA. Okay, why not just store genomic DNA libraries? But what the advantage is, there are no introns here. Okay. Eukaryotic genes commonly contain introns which are non-coding sequences, and these are removed after mRNA synthesis. So this cDNA does not contain any intron. So a cDNA copy of a gene can be isolated as a single intron-free fragment, right? But prokaryotes anyways don't have introns, so this is not a problem. But in general, cDNA is much more advantageous than the genomic DNA in case of eukaryotes. Definitely, there are you can have more templates because there are multiple copies of mRNA for every copy in the genome. So there are more copies per cell of the sequence of interest. All right. And there can be less background sequence. Why? Because only sequences that have been expressed as mRNA will be present in a cDNA. So there is less background sequence as compared to genomic DNA, which makes it less likely that the designed primers will bind non-specifically, right? So one kind of selection is already happening, that mRNA is getting expressed only from our sequence of interest. So the mRNA for our mRNA will also have the sequence of our interest. Fine. So then that mRNA is used for cDNA synthesis. So the primer designing, which will bind specifically to that particular sequence, that particular gene sequence, will be uh, like more specific, will bind more specifically, okay, without showing like really less non-specific binding. Just a- There's another way called homopolymer tailing. This is also taken from the lecture video. What happens here is, in homopolymer tailing, this method exploits the presence of poly-A tail, which we saw earlier as well in the on the mRNA, and then an oligo-DT primer is synthesized to synthesize the first cDNA strand, and then the mRNA template is degraded. Okay, this is same as what we saw earlier, and degraded, and second strand is synthesized. So basically, an oligo-DT primer is used with mRNA as a template to prepare the first cDNA strand, which is actually synthesized using reverse transcriptase using a pool of DNTPs. And then after this first cDNA synthesis, first strand cDNA synthesis, there is this enzyme called terminal transferase, which is used to add C nucleotide at the 3' end of both the mRNA here as well as the cDNA. Okay. Uh, and then this DNA RNA hybrid is formed. And then there is this like upon formation of this RNA DNA or DNA RNA hybrid, what happens is the RNA is hydrolyzed in an alkaline medium, and what happens is fully the cDNA can be recovered. And then again you have the C sequences added here. Again an oligo DG primer is used with this particular cDNA. Now DG primer is used here, which will um, help in preparing the second strand of DNA with the help of reverse transcriptase and DNTPs. Okay, this is called homopolymer tailing. So we talked about terminal transferase. This particular enzyme, what it does is, um, first the DNA ends are chopped off by exonuclease, which only acts at the terminal phosphatase groups, okay, which removes the, terminal nucleotides. And then what happens is this terminal transferase can actually add one last nucleotide at the terminal ends. Uh, Basically, this is how radioactive labeling also happens. Synthesis of radioactive probes takes place by using this terminal transferase enzyme and the radioactive nucleotide. In this case, it's DATP. So this radioactive nucleotides are added at the 3' end of both the strands, and then this particular probe can be purified after synthesis. Okay. Now, another way to screen out and really isolate your like isolate the cells of interest having your recombinant DNA is colony hybridization. What happens is you replicate colonies on a durable membrane, um, which is usually a nylon membrane, prior to hybridization with a labeled nucleic acid probe. So earlier we saw how probes are synthesized. Now these probes are used to have the colonies. So now the cells that have taken up your recombinant DNA are selected for, and these are grown on Petri plates, okay, on a suitable nutrient medium. Now, this is a nylon membrane. This has bacterial colonies here on the agar medium, okay. The nylon membrane is placed on it. So, however the colonies are placed in whatever way, so the colonies are exactly transferred onto um, the nylon membrane. Now, this particular nylon membrane, this is the plate here, source plate here. You have the nylon membrane, you have the colonies growing on the, all the colonies which have taken up the recombinant DNA are growing on the nutrient medium. Nylon membrane is um, put on it in this way, and then however it is put, exactly the way the cells have, the bacterial colonies are there, uh, these will be replicated. I mean, these will have some cells sticking from each colony, and then the same way the nylon membrane is taken up and then put onto a very fresh nutrient agar medium. Okay, so what will happen is, you invert the membrane and lay over new agar surface and help the bacterial colonies to regenerate on top of the filter. Okay, because it will definitely have a few cells from each colonies on the membrane. And then what you do is, it is taking up the nutrient from there, growing, and so it will have a replica of all the bacterial colonies on this new agar medium. And then what you do is you remove the membrane. The membrane is already there. The membrane will have the colonies attached. Now what you do is you soak that or use that membrane for lysing and denaturing the cells, and fix it on a fix it on the membrane. Okay, after lysing the cells. And then suppose you want to, you have designed a radioactive probe against a nucleotide or nucleotide sequence of your interest. Now after lysing the cells, you will have the cellular contents here, and then you probe it using the uh, design probes, and then the probes will bind to those colonies which have the recombinant DNA of your interest. Okay, or which basically have those cells or the colonies which have the genetic sequence of your interest will only be having the probes bound to it. Okay, so this is how you will have the probes bound to it, and then uh, you can use it for further downstream processes to really culture out those colonies, culture those particular cells from that particular colony which is having the probe bound to it. Okay, and then you can use it for plasmid isolation and further downstream processes. Also, earlier one was using, suppose, a radioactive probe here against the DNA of your interest. Now, what also happens is screening by immunological methods. You can use antibodies. Similarly, how you have the master plate, you have the matrix here, the colonies were transferred, and then those were um, grown on the membrane, and then you have lysed the membrane, like you have um, exposed the membrane with the colonies or cells to multiple solutions for lysis, and then now you want to probe the proteins present in the cells. Now you have the bound protein here. The cells have been lysed now. Proteins are bound to the matrix. Now there are antibodies which are specific against the protein of your interest. Okay, so those antibodies are used. Now usually either one antibody can be used or two sets of antibodies can be used, which gives it more specificity and more detection. Okay, which makes it more detectable. What happens is primary antibodies bound are specific to the protein of your interest, and then secondary antibodies bind to the primary antibody. Okay, so the detection is, the signal is amplified. All right, now suppose this is the protein of your interest which has, where the antibodies are bound, and then you what happens is you now have the colony of your interest here. Okay, now you know that this is like, out of this was replicated, right? So you know the position of this particular colony which is positive for your biomolecule of interest, which is expressing your protein of interest or having the genetic sequence of your interest. Then what happens is you just pick it up. Suppose this is the colony here. You just pick it up and then use it for further processes for culturing and then plasmid isolation. Okay, and there's screening by enzymatic methods. This is based on the ability of protein to exhibit an enzymatic activity. This method is not very specific, but allow us to identify a class of protein with known enzymatic activity. Okay, now suppose if, how we saw earlier, that proteins are bound to the matrix. So suppose if these proteins were able to exhibit some enzymatic activity, then the substrate can be put there, and the um, suppose substrate, when this enzyme acts on a particular substrate, it breaks, it lead produces a colorigenic product. Okay, something that shows color. And so what happens is, if that color is coming off, then that means this particular colony has that protein of interest that is showing the enzymatic activity. Okay. Now you have to isolate the gene. Now once the position of the clone is known, it is extracted from the master plate and the plasmid is isolated. Okay. In some cases, clone is further diluted to check the homogeneity of the clone, and purity and presence of the clone is further tested with a PCR using sequence specific primers. Okay. Now basically the gene is synthesized or amplified using specific primers. So if you have ideally isolated after colony hybridization the particular gene of interest, then it should be able to uh, amplify using the specific set of primers which are specific to that particular gene sequence. Okay. Now, how do you really isolate a plasmid DNA? Okay. So plasmid DNA extraction, again, for taking out plasmid DNA, it is an intracellular content. So you have to break the cells open. What you do is, um, you have to lyse it, definitely. So you have to lyse it. Also, plasmid DNA is not part of the genomic DNA. Okay. Plasmid DNA are and plasmid DNA extraction is actually trickier because it should be kept separate from the genomic DNA, which is also written as gDNA, to avoid cross contamination. All right. Now lysis, what happens? Lysis is more subtle than simply digesting the cell wall with enzymes or bashing it with glass beads. What happens here is lysis buffer. In the case of plasmid DNA extraction, we use sodium hydroxide and a detergent, which is SDS, which completely denatures the plasmid DNA as well as the genomic DNA. Basically separates the DNA into single strands. This is very important to perform quickly. We have to be really quick in this step, because if we denature it excessively, then what happens is it can lead to permanently denatured plasmid. The plasmid can't really recircularize, okay, or like renature. And then what you do is, after lysing, you have to neutralize. There is this neutralization step where potassium acetate is added to renature the plasmid. And this helps in separation of plasmid and genomic DNA. Why? Because plasmids are really small as compared to genomic DNA. So plasmids can easily reanneal and renature, thereby forming double-stranded DNA. But genomic DNA are really heavy and large, which takes too long to reanneal fully. So what also happens, the genomic DNA tends to tangle. Okay, so the complementary strands actually remain separated. And then when you centrifuge it, what happens is gDNA, usually bound to different kinds of proteins, pellet um, down. It actually pellets down and comes at the bottom of the tube, and plasmid DNA usually stays in the solution. Okay. Now, at this point, we shouldn't be vortexing or mixing the sample vigorously, because gDNA can break easily. What we saw earlier as well, because gDNA can break easily, that's why we have to be really gentle during gDNA preparation also. And or also while gDNA breaks easily, so that's why while isolating plasmid, while separating plasmid DNA from the genomic DNA, we have to be uh, little gentle. We shouldn't be vigorously mixing it or vortexing it, because what can happen is the smaller, because it breaks easily, the smaller genomic DNA fragments can actually contaminate your plasmid DNA extraction. Okay, why? Because now the gDNAs what particular characteristic are we using in this method is that gDNA is too large. It is taking a longer time to reanneal. Okay, uh, to reanneal fully. So that's why we don't want the gDNA to form smaller fragments, which can actually, again, because it's smaller, it can reanneal and then come along with the plasmid DNA in the aqueous phase. Okay. So finally, when gDNA goes into the pellet and plasmid DNA comes to the into the solution, you have to purify the plasmid DNA. You have to very carefully take the supernatant out, like we saw in the phenol chloroform extraction. You have to very carefully take the plasmid DNA out. You shouldn't be taking it very forcefully, because the pellet, the genomic DNA pellet, can also come off along with your aqueous solution. And also, we should be careful that we don't touch the tip of the pipette. Okay, I mean the microtip. Uh, it will also contaminate your plasmid DNA sample. And then plasmid DNA in the supernatant can be precipitated with ethanol or cleaned up using phenol chloroform or a spin filter. Okay. Do we have the spin filter here? No. So what happens here is um, like either you can use phenol chloroform, what we saw earlier, that DNA will eventually come to the into the aqueous phase, and other contaminants will move to the interphase or the phenol phase, or it can be cleaned up in that way, or the plasmid DNA can then be purified plasmid DNA can be precipitated. How do you precipitate it? Using ethanol. Okay, so what actually happens is, in ethanol, usually in water, the dielectric constant of dielectric constant of water is like really high. So the forces between the ions, if it's a coulombic force between the ions, it's actually inversely proportional, right? So if it's higher, the force is like really low. So they are separated out. Now, in the presence of ethanol, which has a much lower dielectric constant, what happens is the lower dielectric constant helps in more coulombic force between the ions. Okay. So what happens is, usually during precipitation, salts are used. DNA is negatively charged. In salts, you have a positively charged, like sodium acetate is used. Sodium is positively charged, acetate is negatively charged ion, right? So, DNA being negatively charged due to the phosphate groups in it, the sodium ions actually helps like come close with the DNA molecules and um, forms an interaction, electrostatic interaction, and that helps in um, putting the DNA in there. Okay, and it helps precipitate out the DNA from the aqueous phase. Okay, um, yeah. Then spin filter, or many times what happens is there are membranes. Okay, filter columns, which helps in the DNA binding. Okay, so due to the certain buffer salts that are present, which are guanidine salts here. So what happens is neutralization buffer in this scenario contains some guanidine salts, so that the lysate that has been applied through the filter on the filter can bind the silica. This membrane, the spin filter membrane, has silica membrane. It can bind directly for further washing and elution, and the resulting DNA can finally be eluted out, which is pure enough for most of the downstream molecular biology applications. Plasmid DNA isolation is compatible with mammalian and other eukaryotic plasmids. The method that we saw earlier uh, is actually compatible with mammalian and other eukaryotic plasmids, and also small extra chromosomal DNA species as well. So the plasmid DNA is typically between 3 to 5 kb depending on the insert size, and the specific origin of replication present will influence the plasmid copy number per cell. A typical high copy plasmid such as PUC or P-blue script yields around 4 to 5 micrograms of DNA per ml of culture. Okay, plasmid, so one plasmid DNA is there. Now, to make multiple copies, it also has to have one origin of replication, right? For the plasmid DNA to replicate. So, depending on What type of origin of replication is there? The plasmids can be high copy number as well as low copy number. To isolate high yields of plasmid DNA, usually we use cultures in the late log phase or early stationary phase. We looked at the bacterial cultures growth curves, right? So usually to have high yields of plasmid DNA, uh, towards the end of the log phase, we usually harvest the plasmids. We prepare cultures using fresh single colonies and fresh selection antibiotics at the right concentration for plasmid maintenance. And it is important not to overgrow bacterial cultures because this may result in gDNA contamination in the plasmid extract. Okay. Now, very briefly, what happens in DNA replication? It's semi-conservative in nature. It starts at the origin of replication that we saw just in the previous slide, and it is synthesized by DNA polymerase in the 5' to 3' directional. It can be unidirectional or bidirectional, and it is semi-discontinuous in nature, having one leading strand and one lagging strand, and RNA primers are required. Okay. So DNA replication is a three-step event. Initiation, which prepares the DNA for synthesis. So specific proteins bind to the DNA. The double helix structure of the DNA has to be opened up, because there are proteins which are also mostly enzymatic in nature, also helps like helicases to open up the DNA helix. Then there's elongation, which is the synthesis phase where proteins connect the correct sequences of nucleotide proteins, basically the enzyme, connect the correct sequences of nucleotides in a continuous new strand of DNA. And then there's termination, where the DNA synthesis stops again due to mostly the action of different kinds of proteins. So helicases separates the two strands of DNA. Primase helps in the synthesis of RNA primer because that's how the DNA synthesis starts. Polymerization of DNA starts. Then there's single-strand binding proteins, which prevents re-annealing of single strands. DNA polymerase synthesizes a new DNA strand, helps in joining of the nucleotides one after the other. Then there's tethering protein, which stabilizes the polymerase, right? Polymerase has to be held in place also. And then there's a joining protein or joining enzyme, which is the DNA ligase, which helps in um, joining the two ends via phosphodiester linkage. Okay. Now, we have been talking multiple times. I have mentioned about amplification of the gene of interest. So the technique used is polymerase chain reaction. What actually happens is DNA is double-stranded. So you have to initially denature it. It melts the double-stranded DNA into single strands. You have the hydrogen bonds is like dissolved here. And then what happens is you have specific primers designed specifically for the particular sequence of DNA. So these primers bind, okay, from at the 3' end. Basically, the polymerization will take place from the 5' to 3' end, right? So it will bind at these 3' ends, and which is called annealing of primers. Usually denaturation takes place at much higher temperatures of around 95 degrees Celsius to 98 degrees Celsius, and then the temperature has to be lowered for the primers to join, otherwise the primers will eventually join due to complementary hydrogen bond formation only, right? Mostly due to hydrogen bond formation. So if the temperature is still high, it won't let the primers anneal onto the specific sequence. So the temperatures have to be lowered uh, to like to let the annealing process happen. Then finally the polymerase enzyme helps in, and again one kind of enzyme is Taq polymerase, which is a thermostable enzyme, because the thing is the whole solution is put inside, and then the different cycles of temperature goes on. Now it is going, the temperature is going up to 95, 98 degrees Celsius during denaturing. So the polymerase enzyme that is there, we talked about some proteins which are like many proteins which are really sensitive to high temperatures, okay, which denature at high temperatures. So the enzymes are basically proteins only. So the polymerase that will eventually help in elongating the or polymerizing the DNA strand has to be definitely stable at such high temperatures. So usually a thermostable DNA polymerase is used uh, to elongate the DNA molecule, DNA strand, or to synthesize a daughter strand. And then after elongation, the duplication happens, and this is how multiple cycles lead to millions of copies of the genetic sequence of our interest, okay, or we also call it as PCR product. All right, denaturation, annealing, elongation, this cycle goes on uh, according to the requirement of the like, researcher. What happens is you amplify the DNA and then uh, have it in exponential copies, okay, millions of copies. Now again, this is a brief history of polymerase chain reaction. And like in 1950, it was the mechanism of DNA replication was discovered by Kornberg. Okay, he actually discovered the first DNA polymerases and different enzymes like helicases and as well as factors like primers. Then again, quite a few, like 2 decades later, or approximately 3 decades later, you have isolation of thermostable DNA polymerase from Thermus aquaticus. Thermus aquaticus is a um, bacteria which grows in thermal vents, like at really high, it can thrive at really high temperatures. So if it is able, if that organism is able to thrive at really high temperatures, so that means all the enzymes that are inside to help in the different metabolic pathways inside that particular um, thermal like inside that particular bacterium, that can be actually isolated and harvested so as to use in this particular so as to use in some other application, okay. So this is how the DNA polymerase, which is thermostable, was isolated from this particular organism, bacteria, which grew in thermal vents. And then Mullis discovered DNA oligoprobes, and 1983, the same year, repeated thermal cycling was used to create a small segment. I mean, repeated thermal cycling was used to amplify a small segment of gene, and then it was patented in 1985 for PCR. 1985, thermostable DNA polymerase was used, and only at that time TAC, like it, the name comes from Thermus aquaticus, TAC and BST known at the time. TAC was found to be more suitable, and then the development of this thermal cycler, which is the instrument to carry out PCR, was developed, and different kinds of reagents were developed. So earlier, what used to, so in general, what we saw for a PCR reaction to occur, you need to have DNTPs, you need to have the template, you need to have proper ions, okay, proper buffering agent as well as ions for the polymerase to really perform its function, and then you need to have the DNTPs definitely. So earlier, individually, all these reagents were added for the PCR reaction in the PCR reaction mix. Okay, but slowly nowadays, what happens is already a master mix comes industrially, commercially available master mix is there, where already the enzymes, buffer, salts, um, ions, etc., DNTPs, everything is added. You just have to add your primer, your template, and the master mix of a particular concentration is there. So you have to dilute it and add it accordingly, and then you put in your reaction. Okay, so it has become quite easier now, comparatively easier now. Now, when we talk about primers, um, primer length has to be primers are basically oligonucleotides of 18 to 24 bases in length, and they should have a particular melting temperatures in the range of 52 to 58 degrees Celsius. It can also have melting temperatures, depending on what is the sequence of your gene of interest, the melting temperature can slightly vary as well. Like it can be different from 52 to 58 degrees Celsius as well, depending on what is the content. So the GC content of the sequence gives a fair indication of the primer TL. Okay. So if the GC content is too high, what happens is the primer, what also can happen is the primers can anneal by themselves, forming a primer dimer. Okay. Now that can eventually give like uh, unwanted bands during your or unwanted amplification during your PCR, and it will also hinder in the amplification of your gene of interest, because it will eventually lower the concentration or quantity of primers available for the gene of interest, available for annealing to the gene of interest, right? And the two primers should also be prepared in a way that the difference in their melting temperature should not be more than a certain range, 2 degrees to 5 degrees Celsius. Otherwise, what will also happen is it will result in poor annealing efficiency. Suppose the forward primer is annealing uh, at a particular temperature and the reverse primer can't really anneal at that temperature. Suppose like it's not able to form proper hydrogen bonds between the bases for complementarity, like for the complementary bases. So that won't really help in this scenario. Okay. And primer annealing temperature shouldn't be too high. It can produce insufficient primer template hybridization. Okay. So that will again have low PCR product yield, while a low primer annealing temperature will lead to non-specific PCR product. Okay. GC content, we talked about it. The number of G's and C residues in the primer um, should be as a percentage of the total bases should be between 40 to 60%. And when we talk about the GC clamp, because GC has 3 hydrogen bonds, AT has 2 hydrogen bonds, right? So as GC forms a stronger bond than AT, the number of GC content at the 3' end of the primer should not be more than 3. Otherwise, what will happen is it will result in a non-specific tight binding at regions where G and C residues are abundant. Dimers I talked about. Okay. Then there can be repeats and runs. Repeats are consecutive occurrence of dinucleotide, whereas runs are continuous stretch of a single nucleotide. And a maximum number of repeats and runs accepted is 4 dinucleotides, suppose AG, 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 and 4 base pairs respectively. Primer template homology. Primers should be designed in such a way that there should be no homology between the template other than the target site. So what will also happen in this scenario is the primers can actually bind to some other template altogether and help amplifying that. Although we don't want that sequence of our interest. Okay, only the sequence of our interest should be amplified. Now, usually primers are for a primer length of less than 14 nucleotides. The melting temperature is usually calculated using this formula. And for a primer length of more than 13 nucleotides, this is the formula used. Okay. This is taken from the lecture video. Now, how do you really, okay, you have performed PCR. Now, how will you really analyze the PCR reaction? To analyze the PCR reaction, we perform something called gel electrophoresis. What happens here is usually it's agarose gel electrophoresis, where you have an agarose gel with cross-linked um, uh, residues of sugar, and then what also happens is it form the cross-linking of these residues help in forming pores, and the DNA being negatively charged, the amplified PCR product being negatively charged, actually passes through those pores, and um, what happens is this is uh, the negative electrode here and positive electrode here. So the DNA is loaded on this part towards the negative electrode. The repulsion helps in moving the negatively charged DNA towards the positively charged um, electrode, and that actually provides the force for the DNA to move from the negative to the positively charged electrode. And but then this force it moves through the pores formed by the cross-linking of those um, polymerized uh, molecules. Okay. So once PCR cycle is completed, the now based on the size, like if the DNA is much larger, it will take more time to move through the pores, whereas smaller molecules can move through the pores very easily, right? So if a smaller molecule um, will be much farther, it will move much farther down the gel, okay, and bigger or larger DNA fragments will stay towards the upper part. So once PCR cycle is completed, the amplified product is loaded on the agarose gel and it is observed using ethidium bromide. Ethidium bromide is a dye which actually intercalates in between the DNA bases and it fluoresces. Okay, it gives out, it can actually be visualized under UV light. Okay, um, this is there. Now, generally, the more percentage of agarose gel means more amount of agarose is there. That means the pores formed, uh, more amount of agarose is there, it's more concentrated. That means the pores are much more regulated and much more smaller. Okay, and the gel is uh, so a higher percentage of agarose gel is used for um, resolving like really small DNA fragments because it can be resolved very nicely through the smaller pores. Now, if the agarose gel percentage is like really less, like 0.5%, that means for the really large molecules of DNA, it will have larger pore size so that the larger DNA molecules can move through the pores and get resolved properly. Okay. Now, applications of PCR can have, like PCR can have different various kinds of applications in plant tissue culture, plant species identification, disease diagnostic test, cloning, definitely cloning, food science, parental testing, criminal investigation, diagnostics like PCR in infectious diseases. So, like you have, this is what was done few years back during COVID testing also, right? It so you will have a particular infectious gene genetic segment there, and then but in order to have it in detectable amounts, detectable quantity, you have to amplify it. So PCR was used, like it has like this term RT-PCR and COVID testing and RT-PCR was um, so commonly on everyone's household and everyone's certificate, right? So this is what was the basis of the technique, okay, where you are basically amplifying a particular gene of interest or genetic segment. It is also used in diagnosing, like how it was used for COVID testing. It is also used for diagnosing HIV, hepatitis, human papilloma viruses, and gonorrhea, tuberculosis, etc. Okay. Also, um, it can be a very powerful technology to detect the diseases for genetic testing as well as for parental testing. Now, suppose if somebody is prone to cancer because it has this candidate gene BRCA, then that BRCA gene, if we PCR amplify it using PCR, then it can be predicted whether a particular person is predisposed to breast cancer or not using this particular technique. So this is a very good tool to for diagnosing or pre-diagnosis of breast cancer. Okay, this is just an example. So this is what I was talking about COVID-19 molecular testing. Like they had the swab taken, RNA was extracted, and then it was amplified. Okay, the cDNA was prepared and then amplified, and then based on the amount of cDNA produced, one could say that okay, whether it's really at the level of developing like COVID, if it's a serious condition or mild condition, etc. Okay, or similarly, the same way using saliva also. Basically, from both the samples, RNA was extracted, followed by cDNA synthesis, and then amplification using PCR. And RT-PCR principles are usually RT-PCR is basically reverse transcriptase PCR. So one-step PCR is in which all the reaction components are mixed in one tube, and uh, it the resulting cDNA cannot be used for detecting multiple messages. Okay, this is just one-step RT-PCR. And two-step RT-PCR, actually one is the RT reverse transcriptase reaction is taking place. Okay, and then you are amplifying the particular cDNA that has been synthesized. Okay, so RNA is first reverse transcribed into cDNA using an enzyme called reverse transcriptase, and the resulting cDNA is used as templates for subsequent PCR amplification using primers specific for your gene of interest. It can also be used for genetic testing. Okay, it has become a powerful tool to detect diseases associated, what we talked about already earlier. Okay, knowledge of disease associated gene in the person predisposed to these disorders that we saw earlier as an example was breast cancer, have a chance to control the problem much in advance. During criminal investigation as well, suppose a sample of blood, hair root, you see the forensic department collecting evidence from criminal sites, right? So what they do is, like, if they have a hair sample or some tissue left, finger like blood sample of blood is left in the crime scene, what can actually happen is the person's DNA, the criminal's DNA can be there, but that particular small amount is not enough. So the DNA is actually amplified using PCR, and then there can be multiple suspects. So from each suspect, their DNA is also taken, and then it is matched. All right. So you see, from the DNA that was amplified from the crime scene, this is the profile of the whole the DNA that was purified, and then this is the profile that is seen on the gel. Now there were four suspects. You see the profile does not match, so this person is innocent. Suspect 2 is also innocent, but the profile of suspect 3 actually matches, whereas suspect 4 also has a mismatched profile. So suspect 3 becomes the convict. Okay. So this is the end of the video, and there are assignment questions based on these topics only. Okay. Uh, so you guys try solving the same, and then the next week we'll come back with the next week's tutorial session. Okay, I will stop presenting.